Hello, everyone. I realize that the title of this video is a bit clickbaity, but I still think it applies pretty well to an issue that I wanted to talk about today, which is trying not to buy the wrong antibody. This is a problem that a lot of researchers encounter, especially if they haven't had to pick their own antibodies from a vendor previously. In particular, people may end up ordering an antibody that looks good for their purpose on paper, but ultimately fails to stain the target in practice. Further, primary antibodies, those that bind directly to the target, are typically quite expensive, running between $200 to $600. So, if you end up buying the wrong one and you're unable to return it, that's definitely an expensive mistake. Let's talk about some of these antibody selection issues. In this presentation, I'll pose some questions for you to answer. When I ask each question, I recommend that you pause the video to try to figure out the answer on your own before I give my answer. And a few disclaimers before we get started. First, most of the figures that you're going to be seeing today were not created by me, so I have put source links next to the respective images. Second, although I will be showing pages for specific antibodies on vendor websites, I do not endorse nor discourage purchase of any of the products that are shown today. I'm not receiving support from any of these vendors. I don't have problems with them. That's that. So, with those disclaimers out of the way, let's get into it. First question: how do antibodies made by a vendor bind to a very specific target? So, generally, antibodies aren't made against an entire whole protein. Instead, vendors will isolate a specific sequence of amino acids from that protein. So a chunk of the protein is isolated, amino acids are selected out of that chunk, even. So if they are making an antibody, they can selectively target a piece of whatever that target is. An example that I'll be using a lot in this presentation is if there's an antibody against dopamine 1 receptor, they won't try to use the entire dopamine receptor. Instead, they might cleave off the last 20 amino acids of the C terminus of the whole sequence of that receptor. And this 20 amino acid strand is then exposed to immune cells that create antibodies against that strand. If the vendor is touting that their antibody is highly specific, such that it does not bind to similar proteins, then they've likely chosen a particular sequence that has little similarity to most other proteins, or at the very least, the proteins that are found in the organ of interest. An example of this might be that a vendor's antibody could be so specific that it targets dopamine 1 receptors, yet it does not target dopamine 2 receptors. A little bit of an explanation about the diagrams you see here. First, we see an example of how an antibody, this Y-shaped structure, also called an immunogammaglobulin, IgG for short, can bind to multiple sites of a larger protein target, the antigen. So the epitopes are these specific pieces of the antigen, the specific pieces of the target that the antibodies recognize and stick onto. Antibodies can choose different epitopes on the same antigen target. So antibody A will always bind to site A and not site B or C. Antibody B will always bind to site B and not A or C, and so on and so forth. In particular, a lot of antibodies rely on these amino acid sequences that make up these epitopes to be in the right order and accessible to the antibody. So even though we could think of a protein being this loop-de-loo of amino acids, the ones that are most important are the ones that are highlighted as these ball shapes here and then drawn down, zoomed in here at the lower portion of this graphic. Next question. On a related note, how are antibodies specific to the protein of one species, and yet those same antibodies do not target the same protein in a different species? So it turns out that even if one species has the same general protein structure for a specific target as another species does, there are small differences in the sequences from one species to another, and this might be enough to prevent an antibody from binding to one of those species. For example, if an antibody is meant to target the human dopamine 1 receptor, it isn't a guarantee that it will actually target the dopamine 1 receptor in mice or rats or cats or bats and so on and so forth. So it really depends heavily on the sequence of that target that the vendor isolates. And if the sequence has enough differences between species, the antibody could work on one species and not the other, even though the same general protein structure is largely the same. Just a few tweaks in the amino acids could make some antibodies not work on a different species. So we also have another example here where we have a comparison of a sequence of amino acids between yeast and Drosophila. And even for where there are similarities, if there are too many differences, then an antibody is not really going to recognize it. The thing is that these amino acids could still cause the proteins to have the same general shape and structure and function. It's just that there are numerous sequences that can yield that result. So the building blocks can be very different, but the end product looks pretty similar between these species. And an antibody will have a hard time trying to bind to one thing and yet still bind to another. So it might recognize yeast for certain sequences here in this protein and not Drosophila. This begs the question, how do I figure out if an antibody might work for a species that the vendor does not specify? A lot of people might be wondering this because let's say somebody had bought the wrong antibody. It was for some sort of target that's in human, but you use rat or mouse tissue. Okay, we could try to figure out if there is a high likelihood of it working aside from just testing it. We could determine if certain antibodies might actually work. And this might be effective for antibodies against very particular targets that let's say these antibodies are not sold or made by most vendors. Like they're very, very rare antibodies. And so you're trying to figure out, will this actually work for my application? Well, we could actually look up the sequences and compare them side by side to see how similar they are and thus get an estimated likelihood that the antibody will actually work from one species to another, even if the vendor has not explicitly tested that themselves. The way that we do this is that we can use protein databases. We can use NIH's, that's National Institute of Health, BLAST-P site. So when I say BLAST, it isn't a guns blasting kind of blast. Instead, BLAST in this case stands for Basic Local Alignment Search Tool. Per the name, we are trying to align multiple sequences and see what the similarities and differences are when they are the same general protein. So I do have a few examples to present you with. First, here's the link to their page. You could also just look up NIH BLAST and then look for protein blast being a specific thing. So we can click on this, bring us to that. You'll notice that it kind of gives you this query sequence. There's a lot of extra things you could enter into here, but if you're not familiar with using it, I don't suggest you worry about all these other little factors. However, you can't just enter something like, let's say, dopamine 1 receptor. It won't know how to process them. You either have to give the exact sequence of amino acids based on their letter designations, or you have to give a specific identifier number. How do you get that identifier number? You can use other sources in order to find it. So let's take a look at what some of those other sources might be. For example, on the Wikipedia page for dopamine receptor D1, that's the same thing as dopamine receptor 1. So D1 is like maybe the more commonly used shorthand for it. We can go to this page. And typically on the right-hand side, it'll give you a bunch of information or informatics data. In particular, we can take the information for the ref-seq protein part. You'll see that there are three numbers, and usually this corresponds to the identifier numbers for this protein, the dopamine 1 receptor, in three different species. Usually these are human, mouse, and rats. The order I'm a little less certain on. I think the first one's almost always human. I'm not sure which one ends up being rat or mouse out of the remaining two. So let's go ahead and just copy this NP number, and then we can go back to the protein blast site and just paste that in there with any sort of spacing that might be copied over. And if we hit blast all the way down to the bottom, it will run it. Now, when you run these database searches, one of the things that you'll have to keep in mind is that it will usually take a little while to actually do the search. This isn't one of these things where it's slow internet or bad Wi-Fi. It actually is running the search, and it can take anywhere from, let's say, a solid minute to a few minutes. So when you're waiting on something like this, I suggest getting up, getting a drink of water to stay hydrated, going to the bathroom, moving around a little bit, and then checking in on it to see if it's done. Here we go. So we scroll down and find out what is it looking up. Description, D1A, dopamine receptor, Homo sapien. So the specific number that we put it in, it automatically recognized as the human dopamine 1 receptor. And it gives us the length that this dopamine receptor is in amino acids. So then we get a whole lot of stuff going on down here. What we can do is, if we have a particular animal in mind, we can click on one of these entries to see what the percentage overlap is and where the overlaps or differences are. The most important columns are the description being, is it dopamine 1 receptor? The scientific name being of the species typically in this column. And then we see the percent identity, so how much overlap there is, and it's a descending order. So we could try to look down to see if we see a familiar animal. Okay, so let's say that we were trying to compare the dopamine receptor of a human with that of a wolf, Canis lupus. So we click on it and we get the alignment of the two things. We've got the dopamine 1 receptor for humans in the top row, the query, versus the dopamine 1 receptor sequence of that in wolf, the subject line. And so we have amino acids 1 through 60, 61 through 120, and so on. The middle row is where there are matches. And this is where we can see if there are differences. So there's a difference where there's a gap here, small difference here, and here. You may see a plus mark. So that's another difference. Generally, there is a lot of similarity. It makes sense because this was registered at about 95-ish% similarity. So as long as an antibody is trying to bind to a stretch of amino acids that don't really differ between the species, like let's say it was specifically this stretch, then it's likely that the antibody that works in humans would also work in wolf, let's say. However, if the antibody were targeting a stretch that actually had multiple differences, then it might be actually quite problematic. For instance, if it were targeting this sequence right here, because there are three differences among this batch of amino acids, then the antibody has a higher probability of not working between human, where it might work, and then wolf, it is less likely to work. Okay, so that's one example. There is another example we could use. There is a totally different database. So let's take a look. Vendor might give you actually the specific sequence. For instance, we have a vendor that tells us about their anti-NMDA R1 antibody. So in this case, it is something that binds to the NMDA receptor, specifically subunit 1. And thankfully, they actually give us what their sequence is, their immunogen, a synthetic peptide within human NMDAR, amino acids AA, numbers 849 through 913. So although they don't give us the exact specific sequence, they say, oh yeah, it's somewhere in the span of these amino acids. It does narrow it down considering that the receptor seems to have a lot of amino acids. Now, this is all well and good, but what about this other database I was talking about? Well, it gives you a little bit more information about the target here. And interestingly, it actually gives you the UniProt number. So if you were to plug in the UniProt number in the UniProt website, it would pull this up. So let's go ahead and pull up that UniProt number through the direct link that they've provided. We have NMDA receptor human. Lovely, great. However, we also want to maybe see what else we can have. So what we can do then is we can run a blast on it. And so if you're trying to figure out what species it's similar to, this will actually bring up a whole list of different species. The amino acid sequence is already loaded, so you're good to go. If you want to cut it down to the amino acids specified by the vendor, you would just delete everything except 849 to 913, let's say. So we could hit this, hit run blast. That's all well and good. Because I've done this before, it actually saves the previous searches. Some have expired since, so that's fine. But let's go ahead and look at the blast sequence from the last time I ran this search in preparation for this video. So it gives us the target, the entry that I used. But also, if we scroll down, we may see some familiar animals. For instance, we got another bear-like thing, fox, cheetah, other such things. We can use the browser's search function in order to find the other things of interest. In particular, what we can do is we can just hit in control and then musculus for mouse. In this case, I happen to be hovering over Rattus norvegicus, so rat. I'm going to just go ahead and show you that if we put in Rattus, it highlights that, jumps straight to it. That's convenient, lovely, great. We're going to hit the checkbox here. We have checked off first option given by the website, and then the next option being Rattus norvegicus. And we're going to ask it to now do an alignment, align selected results. Alternatively, you could just uncheck the thing that we use as a search and then just align selected results with query, basically the same. Let's go ahead, align selected results. And now it's entered two different things into the query box. It will align these for us. And we may need to wait a little while before it pops back something that is useful for us. Again, I did this search previously, so we can jump ahead and see how the alignment ended up working out. Oh, actually, it actually finished this pretty quickly. So let's take a look at what it gave us. All right. So when it lines these two things up, we've got the human variant on top, the rat variant on the bottom. It seems like there are a lot of similarities. We do see that there is a chunk of differences in the initial part of the sequence. So there are like 3 different amino acids right here in the beginning, and that's problematic, let's say. However, we're caring more about the amino acids 849 through 913. So we scroll down here. 849 is probably somewhere right around here, going up to 913, should be somewhere around here. And we see that everything's identical. So if we have an antibody against NMDA receptor by Abcam, let's say, and if it said human, but they didn't show any testing on rat or mouse, we could look at the blast search and the alignment tool to figure out, okay, well, if they gave us a sequence are these the same between the animals? If they're identical, then it's highly likely that the antibody will work in both species, meaning that it'll work on humans the way it's supposed to, and it'll work on other animals that have this exact same sequence. It's never a guarantee, because sometimes weird things can happen with tissue processing that alter the protein shape and make a protein not recognizable to an antibody. These small species differences can matter, but beyond that, it is a high probability that the antibody should work on this other species. Why does it matter what host the antibody comes from? Well, the host of the antibody decides what secondary antibody you will end up using on it. So this is really important because when we're trying to actually label a target, rarely do we ever have the label actually attached to the primary antibody, as shown in the mouse anti-X case, rabbit anti-Y, donkey anti-Z. They do not have a fluorescent label attached. Typically, what's done instead is that a fluorescent label is attached to what's called a secondary antibody or an antibody against antibody. So you can have something like a goat anti-mouse antibody that binds specifically to mouse antibodies to properly label them. If you had a specific situation where you only had goat anti-mouse secondary antibodies, but you had a choice of what primary antibody you could use against antigen X, you're going to want to use a mouse primary antibody. You would not want to use a rabbit primary antibody that happens to target X, because let's say that all you had again for secondary was goat anti-mouse. You would need instead a goat anti-rabbit to target a rabbit antibody. So this can be one of those cost-saving measures where you stick with what your secondaries are, and that determines what your primary antibodies should be. Here's a question that I don't have on the slides here, but do you know the difference between monoclonal versus polyclonal antibodies? So, first one, polyclonal antibodies are derived from taking a target sequence of amino acid. So, whatever your protein target is, you isolate a chunk out of it and you inject it into an animal, much like a vaccine. This causes the immune cells in the animal to create antibodies against it, and the immune fraction of the animal's blood is isolated, purified, and the antibodies specifically are removed from the resulting serum. Now, the name polyclonal indicates that the antibodies come from numerous different immune cells. The thing is that these different immune cells make their own different antibodies. So immune cell A will make antibody A, immune cell B will make antibody B. And so we come back to this diagram here where we see that we have all these different antibodies and they attach to different epitopes on the target. So antibody A will only attach to epitope A on the target, will not attach to epitopes B or C. What happens with polyclonal antibodies is that they're coming from multiple cells, multiple different cells, polyclonal. And what this causes is that we have a diversity of different antibodies. They can bind to different parts of a larger target, the antigen. What this means is that by having a greater diversity of antibodies, there is an increased probability that your target sequence will be labeled versus not. However, there is a potential downside. Because these antibodies will look at all sorts of different sequences of the antigen, some sequences might be similar to stuff found in other proteins. So there is an increased chance of non-specific binding to other possible targets. And so that's one major downside of antibody use in general, that if it's polyclonal, there's chances are that some of the antibodies in the selection might bind to something that is not the proper target. And so even if it doesn't cause a specific binding to things that are not the target, There can also just be general non-specific binding where these primary antibodies end up getting stuck on other parts of the tissue and cause a general increase in the background signal. It kind of drowns out the noise. All right, the other side of the coin, monoclonal antibodies. These are created rather differently. So we still have an antigen being injected into an animal's serum. Usually, this is mouse or rabbit because these are the animals that we can actually isolate the cells from and do this monoclonal setup. So rather than taking the blood and purifying out the antibodies, we actually take the immune cells from the spleen and mix them with myeloma cells, a certain type of cancer cell, in order to make them immortal cells. So they don't have a limited lifespan, they'll just kind of exist. And as long as they exist in the form of these hybridoma cells, that's what they become, they will keep producing antibodies. So we have all these mini antibody producing factories all in little well plates. Now, an important aspect mentioned here in the diagram is that each cell produces its own specific batch of antibodies. So all the antibodies in this well are the same. They're all identical because they're coming from this one cell. The same cell produces all the same antibodies. We go over to the next well. This produces a whole bunch of its own antibodies that are all identical. But these antibodies are different from the first and third well, and so on. So the antibodies are highly specific to each cell. We could find out which cell produces the most effective antibodies and then clone and replicate it in order to produce more of the cells and thus more of the antibodies. So we basically set up like a miniature antibody creation factory that keeps creating the exact same antibody. So that's why we call it monoclonal, because the end product is that it's all the same type of cell, just replicated, the same clone of it. And so it keeps on producing more antibody. And the solution surrounding the cell, the supernatant or culture supernatant, will be chock full of antibodies just floating in that solution. Now, how does this play out? Well, we have a different representation of what's happening with polyclonal versus monoclonal. To reflect on polyclonal again, imagine that a product that is a polyclonal antibody from a vendor will have a collection of, let's say, 3 different antibody types against the antigen of interest: blue, yellow, and green, just arbitrarily colored. And each of these binds to its own specific epitope target on the antigen. However, monoclonals are far more discriminating and specific. So we get a monoclonal product from a vendor. All the antibodies in that vial are the exact same, and they will all bind to the very specific same target or epitope on the target. This means that they will not be binding to the blue or green sites in this diagram as a result. So here's what happens with them. You end up having an antibody that has much less of that non-specific binding where it just binds to other stuff randomly. It's definitely minimized. It's not guaranteed to have zero non-specific binding because sometimes antibodies get stuck on things that they're not even really trying to bind to. However, it's much less non-specific binding than, let's say, polyclonal antibodies. However, the big downside is that the strict specificity of these monoclonal antibodies also decreases the probability that the antibodies will bind to the target. That might sound weird. You may be wondering why the binding probability decreases for this. Well, there are 2 potential reasons. Reason one is because there is less potential target to bind to. We only have 1 site out of the 3 possible that the antibody will be able to bind to because it's so specific. Second reason is that there is not a guarantee that every molecule of the target, every one of these triangles in this example, will be accessible to every molecule of antibody. And this is due to the unpredictable nature of how fixation works on the tissue. So fixation commonly obscures the yellow part of the antigen here, then it's very possible that a lot of the monoclonal antibodies will not really work. If they can't access the epitope, then they're not going to bind, and then you're not going to get staining. So that high specificity can come with the downside of it just not labeling things at all if the conditions aren't right. Next question. Let's say we are looking at a company's product page for their antibody product. What information on that page will help inform us about whether the antibody will or will not work for our specific lab's goals? For example, I've provided this antibody product page. So take a moment, come up with a list of things that a vendor page should have that will help inform you about whether it's an antibody that's right for your purposes or not. And then come back and take a look at this. All right. So my list includes the following. Lacking some of these won't be deal breaker, but other ones are more important. First up, the target species, also known as species reactivity. So this is super important for trying to figure out, will it work in your sample? So if you work with mouse brains, then you want to see, oh, is the target species mouse? Is it a list of multiple species and mouse is in there? If so, then it should work on mouse brain. Also important, we already discussed before, the host species. In order to figure out how to label the primary antibody, we need to know what the host species is, and thus we can choose a secondary antibody that will match with that primary antibody host species. Polyclonal versus monoclonal, we already went over the ins and outs of that. Immunogen or target sequence, we also covered to some degree. This is helpful for trying to figure out specific pieces that are targeted by the antibody. That said, a lot of vendors don't give this specific information, which kind of sucks, but I don't find this to be a deal breaker if you can't get that info. Recommended applications. This is pretty important. So the vendor will usually list a bunch of applications, usually by acronym, and you'll want to make sure that they've tested it for your particular application. There are a ton of antibodies out there that have been tested for Western blot, but not much else. And so if you're trying to do immunoprecipitation or ELISA or immunohistochemistry, IHC, if you look for an antibody and all it has on there is Western blot, it's probably not going to work for these other applications. It's not guaranteed unless the vendor says otherwise, but generally you want to avoid just going with any antibody. You want to see if it has the specific type of setup you use. For me, I care about immunohistochemistry, so I want to see that the recommended application includes IHC or even immunofluorescence, anything close enough to that. Western blot will not do it to me. Immunoprecipitation will not do. ELISA will not do for my specific purposes. Pictures of the antibody staining. If you're just doing Western blots, then you want to see pictures of the Western blots. However, for me, I stain tissue. So I want to see staining of tissue in the vendor's own quality assessment tests. And so the antibody staining in tissue is super important because then I'll have some assurance that it works in fixed tissue. The way that proteins are shaped and structured in fixed tissue, so formaldehyde treated tissue, when compared to what happens with proteins in Western blot, proteins can be vastly different shapes. So an antibody that recognizes things in Western blot is not at all guaranteed to recognize things for that are fixed tissue for IHC. And vice versa can be true as well. If some antibody works for IHC, there could be a chance that it will not work for Western blot. So seeing pictures of actual staining is super important. What is also helpful, but not necessarily a deal breaker if they don't quite have it, is where they show pictures of staining in the tissue type you plan to use. So they might show pictures of, like, I don't know, spleen, liver, muscles, other things. For me, I'm interested in brain tissue. If they don't show pictures of brain tissue staining, but they show other organs tissue staining, that's usually okay for me. Ideally, brain would give me a lot better assurance that would work in the brain, but um, it's not really guaranteed that they're going to go all that direction just to get concentration of the product. So you want to see if they have it concentrated more than normal, so more than 1-to-1 ratio of mass per unit of volume of solution, or they pre-dilute it. Some vendors pre-dilute their antibodies in a 1-to-10 dilution, and so you'll have to take that into account as far as making sure you don't over dilute, because then it won't really work. The storage buffer can be important. So usually they use phosphate buffer saline as the storage buffer with sodium azide at a low concentration. If you need to have things that are azide free or are phosphate free, then you could probably request those from the vendor, but generally they should put this on their product page. Reference info for publications that had previously used this antibody successfully. This one's pretty important too. So even if the vendor doesn't provide this information, you're going to want to see if there's any info out there that others have used this specific antibody product and have used it successfully, especially in applications that are similar to what you use. So if a vendor actually puts this information on their page, that's cool. And if you go to these references and you see, oh yeah, they use it in the same tissue type as I did, in the same way that I did. So if I found brain tissue staining, IHC staining, pictures of it in the publications that they list, that would be very reassuring for me to follow. So let's take a look at this web page for an acetylcholinesterase antibody and see what is covered here. Immediately at the top, it'll tell you what the species reactivity is. So this antibody is supposed to work in human, mouse, and rat tissue. The host is rabbit, so the antibody comes from rabbit. Suggested applications are Western blot, ELISA, IHC, middle one being most important for me. Unconjugated just means it doesn't have like a fluorescent tag on it or anything. And they do provide publication information, so some reference info that use this antibody product, as well as some customers that have submitted direct reviews to this website. We also end up seeing some pictures of Western blots, which, as I mentioned, I personally don't care about, but you may find useful. And then we actually see tissue staining, human liver, and more human liver. So it's all well and good. Tested applications, positive IHC tested in human liver tissue. They do suggest antigen retrieval, which I'll talk about later. They have recommended dilutions, the actual applications published. Scrolling down a little bit further, it does give you a little bit more info. For instance, the immunogen being a specific product that they actually sell. Other info as we continue going down, the storage buffer solution, suggested information, etc., etc. Next question. If a vendor states that an antibody is approved for use in immunohistochemistry, yet they do not provide a photo of immunohistochemical staining, should you trust their recommendations? I say generally no. This isn't to say that the vendor is lying necessarily, but it also might imply that the vendor did not test their antibody for that specific format, in particular, in this case, for IHC. So, since my focus is IHC, I've made it very clear that I want to see pictures of IHC stained tissue. I also made it clear earlier that I exclude Western blots as photos because those don't really tell me anything about how their antibody will interact with fixed tissue. So these Western blots, because I don't do Western blotting typically, I don't find the Western blot pictures to be particularly useful. It'll tell me that the antibody works to target, some protein of some size, and they could put in things in solution to block its binding, but it doesn't tell me how it'll behave on a tissue slice, and that is a problem. What I also find helpful sometimes is that vendors actually provide a list of applications that their product, their antibody product, is not recommended for. For instance, take a look at these. I found this on one website where they were saying, oh, yeah, our antibody is not suitable for flow cytometry. immunocytochemistry slash immunofluorescence, uh, immunohistochemistry on frozen sections, or immunoprecipitation. So I find it ultra helpful because it'll save the trouble of even trying to do it in cases where the vendor themselves have said, yeah, we tried it, it doesn't work. You might want to try something else. Two questions here. Do you know the difference between cell culture photos versus tissue section photos on a vendor website? And another question that will follow up with that in a little bit: is your target localized to specific cell compartments or not? These questions are definitely based on your own knowledge and background. However, what I'd like to add to this is that pictures can sometimes be quite ambiguous. For example, I had seen a picture of a dense cluster of BDNF positive cells in a vendor's photo. BDNF stands for brain derived neurotrophic factor, and it is a key signaling molecule used by the brain, but also other organs, in order to help neuron growth, among other things. Let's take a look at the web page real quick. So it gives us the usual stuff, and we do see photos right here at the top. We click here, tells us that they did immunohistochemistry on human colon cancer. Okay, that's all well and good. Immunohistochemistry on human breast cancer. Again, all well and good. That's fine. Immunohistochemistry on human placenta. Sure, great. All right, this is the picture that I saw, but I was not paying close enough attention to the caption. So what I saw here was, oh, there's a whole clusters of cells that are BDNF positive. And so I thought, well, maybe if I stained for BDNF in the brain, I would actually see neuron cell bodies that could be counted or something similar to that. What I did not see in the description, or what I didn't pay attention to, I should say, is that it was done on these U2OS cells. So these designations, these letter number designations, are usually cancer cell lines or specific cell culture that is able to be immortalized and constantly replicate. We see something similar here, where I see more fluorescent photos indicating cell bodies, but these are, again, another sort of cell line in isolation. This is an in vitro rather than in slice U-251 mg cells. So not super helpful. And then Western blots. So the reason why these aren't super helpful is that it gives me the impression that the BDNF stain should be localized to an intracellular space. The problem is that that is not how it works in the brain. And I didn't think about this until after we had ordered the antibody for it. So we had failed to visualize individual neurons after attempting to stain with an anti-BDNF antibody. Again, this is not to say that the antibody doesn't work. It's to say that what we're staining for is not going to be looking like these pictures. I looked into it a little bit further. I first saw that these pictures were for in vitro situations, things that are not neurons, not in tissue. And additionally, it dawned on me, of course, BDNF isn't going to be just stuck inside neuron cell bodies. It is going to be released from cells. It's probably going to be in much higher concentrations outside of cells in the extracellular space compared to inside neurons. So trying to have some sort of very clear contrast cellular staining with BDNF was not something that I should have expected. This, again, to be clear, was my own mistake and not the vendor's fault. But this should also stand as an example to you all about how you should find pictures of staining with an exact type of tissue or with a specific staining pattern that you would expect before you go in and order an antibody. So this brings up another one of the questions I had here. The other question I had was, is your target localized to specific cell compartments or not? Again, might be based on your general background knowledge, what you're trying to stain for, what is your target. But nonetheless, it's always important to do your own background research in the literature to figure out what a typical stain of your target will look like. If most stains are intracellular, then you should expect that your stain with a new antibody you order should look the same. However, if the target molecule is typically found in an extracellular environment, and if it also isn't typically anchored to anything, much like BDNF that I mentioned before, then you might not find pictures of cellular staining in the research literature. And likely you also won't see it in your attempts to stain tissue sections. For neurons, there's another tricky case. This might apply to other cell types, but this in particular applies to neurons based on how they can be actually quite large, just not in the way that most people expect. When people work in cell biology and things like that, when you say, oh, yeah, the cell is large, they expect, oh, yeah, what's the diameter of the cell or things like that. Neuron cells aren't large because of their cell body diameter, but because of the projections they send out over a long distance, the axons, the terminals. So the cell body could be like anywhere from like 20 to 40 micrometers, but the axon could travel several millimeters, even centimeters across a whole brain. And so this gets tricky where certain substances in those neurons might actually be trafficked along the axons and not stay within the cell body. So, in specific, neurotransmitters and peptides are typically, depending on the peptide, not found in the cell body, but instead mainly localized along the neuron's long axons or even all the way at the terminals. What this means is that the target staining could end up millimeters or even centimeters away from the originating cell body, making the attempt to tie a certain neuron's chemotype, what kind of neurotransmitters it uses, or other sorts of identifier information, really hard to pin down if you can't actually stain the neuron's cell body and you just see a bunch of stained fibers somewhere else. There are some solutions to this issue, some ways to resolve it. A lot of them are complicated or problematic. The easiest solution I recommend is that instead of using immunohistochemistry, you use in situ hybridization to stain for the mRNA responsible for creating those peptides or neurotransmitters. This is because mRNA, in more often cases, tends to stick around where the cell body is rather than being trafficked all the way down the axon and the terminus. So let's give an example of what I'm talking about. This first example shows axonal or punctate staining, where even though you might see certain blobs aggregating together, these are not cell bodies. None of these are cell bodies. So what we're looking at are clumps of terminations or axons going through here that are positive for a opioid used by the brain, Lu and Keflin. So we can't really count cells in this area of the brain. It happens to be paraventricular hypothalamus. Another example, if we want to stain for the much more common neurotransmitter GABA, what will happen is that it will stain all sorts of processes all around the brain, but it generally will not stain the cell bodies again. So this makes it hard to tie things down as far as, okay, where are the GABA sending cell bodies? Well, we don't know. Can't really stain for them because the GABA leaves the cell body quite quickly. Or, in fact, in some cases, it's not even manufactured in the cell body to be trafficked anywhere. It might be manufactured in the terminals. So there might be another way of addressing this. You could try to stain for the enzymes that create neurotransmitters, especially if those enzymes tend to be cell body localized. A good example is GAD67. It is the enzyme that makes GABA, well, one of two major enzymes in any case. GAD67 typically is more often located towards the cell body rather than the axons or terminals. Is it not a guarantee that it is only in the cell body? You will find it in axons and terminals, but you will still get very clear somatic staining like you do in this third picture here. So this can be very reassuring for trying to identify GABAergic neurons because you can actually see the cells rather than having a bunch of dots and lines going everywhere that you can't really tie to anything. Next question. Should I perform antigen retrieval if the vendor suggests it? To that I say, maybe? So antigen retrieval can take a variety of forms. This is an old school photo of how some people might have done antigen retrieval. Things are a little bit more standardized, but basically one way of doing it is by using a acidified buffer and boiling the specimens on slides inside that solution on a hot plate, hopefully having it stirring as well. So the thing about antigen retrieval that I don't like is that it can really rough up the tissue and kind of just destroy a lot of things that are important to the process. Now, that said, some vendors are saying, well, in order to get the staining that we got, you have to use antigen retrieval. So if you're planning to use the exact same type of tissue, the exact same tissue prep, and replicate their staining, you should probably follow the protocol as best you can. I do recommend, at least in your pilot testing of a new antibody, doing without antigen retrieval just to make your life easier, because antigen retrieval is always sucky to set up unless you have like a dedicated device to do it. And if you don't need to do it, I really recommend not doing it. And again, there are some specific professions and certain ways of prepping tissue that commonly utilize antigen retrieval. An example would be that I often see paraffin embedded tissue undergo antigen retrieval, where they try to undo all the fixation to liberate binding sites for antibodies to find and bind to, whereas the formaldehyde was left to continue being cross-linking all the proteins. The antibodies can't bind to anything. The problem with undoing fixation is that it makes the tissue weaker. And some of these antigen retrieval techniques, particularly the boiling variety, can be quite harsh on the tissue, especially if it's not the paraffin embedded variety. I also especially don't recommend performing antigen retrieval on free floating tissue. It deforms it a lot. I know this from experience. Um, Lastly, the boiling buffer variants of retrieval can be tricky to set up in a way that they are effective, consistent, and safe to do without injuring anybody. There are other ways of doing antigen retrieval using digestive enzymes, but those come with their own downsides, uh, such as being potentially caustic or over digesting tissue, so that it just turns to mush. So, with that all said, though, if you find that an antibody does not work when you're normally applying it for your own applications, it's worth at least giving antigen retrieval a try if you have the time and the resources and the setup to do it. Next question. If a company that we buy antibodies from suggests that we use a 1 to 100 dilution, that is, diluting the stock by a factor of 100 with some sort of diluent, should we trust their suggestion? Why or why not? Well, I tend to say not always. This is not a blanket no, but it is not a yes. You should always do your own testing. I don't mean to instill any sort of paranoia or imply that the vendor is lying when they say use it at a 1 to 100 dilution. Um, However, you cannot guarantee that their setup for testing the antibody will be the exact same as your setup. Thus, you really have to do a dilution test on your own of the antibody when you get it ordered. The vendors even may say on their page, such as we see in this screenshot here, optimal dilutions should be determined by the user. Now, for anybody who might find it useful, I have an explanation of how to set up a dilution curve test in my IHC lab manual, which you can find via my website. The link is in the description for this video. The lab manual is free to download or look at, so no biggie there. Putting aside the need to do your own testing, it is still possible that the vendors can be a bit misleading. There are profit incentives, and so they may be telling you to use a very high concentration of an antibody in solution just so you go through it faster, you use it up more quickly, you have to reorder it, and that makes them more money. So it may also be a cost saver to at least check what the dilution is that works best for you. Because if you could stretch it a little thinner, like instead of using 1 to 100 dilution, if you work better at a 1 to 1000 dilution, you're probably going to have a lot more to use and you're not going to have to order more and spend more money. Next question. When a secondary antibody product is listed by the vendor as cross-adsorbed, what does that mean and why is it important? Antibodies that are cross-absorbed might also be specified as minimal cross-species reactivity. This is to say that the antibody is only supposed to bind to the intended target animal and not to other animal antibodies. So this situation is particularly important if you're employing multiple IHC labeling. Let me define exactly what multiple labeling means. So we come back to this figure. It's a little bit changed in this case. Multiple labeling involves when you have at least two different targets in the same tissue. So you have the same tissue section and you want to perform two different IHC stains on it. Let's say that you want to employ an IHC procedure that stains dopamine 1 receptors in green and GABA B receptors in red. Let's say in that case, the dopamine receptor antibody comes from mouse and the GABA B receptor antibody comes from rabbit. So we have two different primary antibodies, one from each host species. We need two secondary antibodies that are used to target those primaries: donkey anti-mouse antibody and a donkey anti-rabbit antibody. And each of these secondary antibodies should have different colors attached. But let's pose a weird hypothetical. Let's suppose that after doing all this, what if your donkey anti-rabbit antibody also sometimes binds to mouse primary antibodies? This is a problem. This is some cross reactivity, some cross labeling. This would cause us to mistakenly think, based on false labeling, that dopamine receptors exist where instead they should be just GABA B receptors. Recall what I mentioned before about sometimes antibodies working against things that are the target protein in the wrong species. If an antibody's effects are too broad, it can start binding to things that it's not supposed to bind to. So this applies to secondary antibodies as much as it does to primary antibodies. To kind of explain this again a little bit, we have this example where we do have antibodies that are cross-adsorbed. We have a goat anti-mouse secondary antibody, IgG. In this case, they mention, oh, it's been absorbed against sheep, rabbit, and donkey, meaning that this particular product here will not bind to sheep, rabbit, or donkey primary antibodies that happen to be on the tissue or in solution or anything else. It will ignore them and only zero in on mouse. We have the converse of all these, where we have a goat anti-rabbit antibody absorbed against these other species, won't bind to them, only to rabbit. Same thing with an anti-donkey antibody, only finds donkey, ignores sheep, mouse, rabbit. And we do have an example of this here, where a website tells you, oh, it has minimized cross-reactivity. Sometimes they'll have like shorthand up here, but they do mention in more long lengthened terms that if we have a donkey anti-rabbit antibody, and it says Psi3 because that's the red fluorescent label they've attached to the secondary antibody. Affinipure means that it's their specific product name by this company for it's a pure affinity just to rabbit. It's not going to have reactivity to these other things. So minimal cross-reactivity to bovine, it's cow, chicken, goat, guinea pig, Syrian hamster, horse, human, mouse, rat, sheep, all those other animals. So it's only going to zero in on rabbit. So if you have other primaries attached to the species that you're working with, so if you have rat brain tissue and you're using cow antibody or chicken antibody, this secondary will ignore those. It will only find rabbit and bind to that. And that is a good thing. Because I have experience when you don't do the cross-adsorbed secondary antibodies, you can get that false positive double labeling. And it's something to really watch out for. Next question. Is there a collective database that has info on most antibody products available rather than just doing normal web searches on your own. Turns out, yes, and there are a few. The one that I'm most aware of is antibodyregistry.org. So let's go ahead and take that one for a spin. Here, we can type in what our target is. Even though we can type in a catalog number, like if there's a specific product number that we want information on, we can type in other things. Although I have in my script dopamine 1 receptor antibody, I'm going to try something a little bit different. I'm going to say GAD67 just to see what it brings us. And so what it'll do is it'll give us a whole slew of results of the antibodies, the vendors that sell them, the specific host that is created in, all sorts of other stuff like that. Sometimes they will include reference information. For instance, thankfully, this one actually has reference info from using this particular antibody. And it'll direct you right to the PubMed page, which gives you the abstract for the study. And if you're lucky, you'll have free text access here, which is kind of cool. So if we look at this one, it says that it has use in all these other situations. Basically, though, this can point us in the direction of finding an antibody that will work for our purposes. We can weed out things based on, oh, it's not the right host species. So if it's not the right host species, if we don't want rabbit, then we ignore all the rabbit entries. Oh, it doesn't come with any previous history of use. Well, okay, maybe we narrow down things further than that. Or, oh, we find this paper. Do they have pictures of GAD67 staining that is cellularly located? Let's just take a quick look. I don't have this scripted, so we're just going to find out if there are pictures of GAD67 cell bodies in here. No guarantees, but they'll at least tell you whether they had something or not. So they have GABA receptors, CRF, various other things. No dice on that, it seems. But still, this is how you can find more information from this antibody search database. So there probably are other ones. And if you know of other ones that actually you prefer more, feel free to leave a comment for this video so that people are aware of it and even I could look into it a little bit further. Where can one find cheaper antibodies? This question is a big one that a lot of people want the answer to because uh, a lot of us don't have money piled up in our lab cabinets. A lot of us have to deal with much more slim budgets in order to purchase our research chemicals. Lucky for us, there are some options. There are two options in particular I know of, and these are nonprofits. So nonprofits sometimes are repositories that maintain antibody stocks and the cell lines that produce those antibodies. So those hybridomas that produce monoclonal antibodies, like I discussed earlier in this presentation. These nonprofit organizations are government funded, and as a result, they're kept afloat not by trying to charge the consumer, but by taxpayer money and grants. They also charge a lot less for researchers compared to commercial entities. So if you're working at a university or a college, a nonprofit institution, you can end up purchasing these things for extremely cheap compared to what a lot of other vendors will sell antibodies for. So the two main ones I'm aware of from over the years are the NeuroMab facility, that's neuro monoclonal antibody at UC Davis, and also the Developmental Studies Hybridoma Bank, DSHB, over in Iowa. Let's take a quick look at both of these. So this is the front page of NeuroMab, and it shows us a bunch of random information. If we looked at NeuroMab's tab up here, it actually gives us their full list. And if anything seems to be cut off at the bottom of the screen because of a weird browser error, you can just look at it via the downloadable CSV file to get the full listing. But basically, they will give you information on these antibodies. Some of these PDFs have Western blots. Some of the PDFs actually have images of stained brain tissue employing these antibodies. So very useful. Um, this one's focused on like brain related markers. So if you work in other tissues, you may not find this quite as useful, but it is still a very lengthy list of things. And you can find things that are somewhat related to the brain, but are definitely relevant in, let's say, cancer research like CFOS. So it's all well and good. Let's take a look at DSHB. So this one also has a lot of stuff going on here. Let's go ahead and look at CFOS on here. DSHB has a bunch of hits that come up for CFOS. Let's look at proto-oncogene CFOS. And here's where we get the price tag. Look at that. Most of the time when you try to look for a CFOS antibody being sold by another vendor, whether it be Sigma Aldrich, Millipore Sigma, Thermo Fisher, whatever their names are these days, because it always is changing from buyouts and mergers, they're usually charging like $300+, maybe up to like 500 or 600 in some extreme cases. The stuff that you get from the hybridoma bank here can be $45 for a usable amount. So you may be wondering, okay, what is a usable amount? You'll notice that the product comes in a few forms. You could get frozen cells or growing cells so you can have your own antibody production factory in your own lab. That's kind of neat that they do that. For most people, though, they'll be fine taking the supernatant, which I mentioned with monoclonal antibodies, is the solution that's surrounding the hybridoma that produces these antibodies. So the solution is not like pure antibody, but it's still a high enough concentration, like between 1 to 10 and 1 to 20 dilution. So looking at the volume being a 1 ml volume, 1 to 10, 1 to 20, we end up basically having the same rough amount of antibody to use as what a typical vendor would sell. Like again, Sigma, Millipore, Fisher, all those. What I'm basically saying is that we are getting the same amount of antibody and probably the same quality for 1/10 the cost from nonprofits such as this. So I strongly say that you consider options such as the Developmental Studies Hybridoma Bank and NeuroMab. They also give you plenty of extra information down here that you could look into further to see what's relevant to you. All right. There is a little bit more to be said about this, however. Um, in particular, I want to talk about NeuroMab. I find it interesting that in the last 5 years or so, the Developmental Studies Hybridoma Bank has begun distributing some of NeuroMab's antibodies. So one nonprofit is distributing the antibodies of another nonprofit's. This is fine. I have nothing against that idea. I did notice that with NeuroMab specifically, when they were not able to distribute their stuff via DSHB, they had previously tried to partner with various types of biological science companies. The problem was that although NeuroMab had always charged very low amounts for their antibodies, like less than $100 easily, probably a lot cheaper if they had certain things on sale, they were no longer able to distribute their own stuff at some point for reasons that I'm not clear on and I don't blame them about. But then they partner with some companies to distribute. They had no control, however, over what the companies would charge. And what would happen is that these companies would turn around and say, oh, yeah, this antibody costs $350. Meanwhile, NeuroMab had, up until that recent point, sold it for $50. So these companies would charge a massive markup, and it'd be worse than any other vendor because these companies weren't actually the ones developing the antibodies. Somebody else was already doing the work. So they were basically just using some particularly egregious profit racketeering practice that I always kind of hated. Um, and apologies if some people are involved in this, and this sounds awfully harsh, but I just think that it's really overcharging for like work that the vendors, the distributors didn't actually do themselves. Um, so I don't know what the switch was that caused NeuroMab to do it. I know that they did put out a statement years ago talking about like, we know that you're upset about like the high costs. We're looking for other options for distributors that aren't going to charge this much because we never wanted it to be this way. So again, this is not their fault, I assume. I assume that they want to keep it inexpensive, um, because we want to be able to do research without having to like pay insane amounts of money to make it happen. Um, if there are other nonprofits that you're aware of that distribute antibodies, please do let me know in the comments or email me. Uh, Putting in the comments lets other people know about the options. I think that is always good to just have more options. There is another possibility. If nonprofits don't have the exact antibody you're looking for, commercial vendors may either provide samples or have a lower cost version of their antibody product. Now, the samples thing, you could probably only do so many times for a given vendor, but they do want you to buy certain things. So if they give you a tester sample and it works great, then that's incentive for you to buy it and they might make money from it. So they don't really lose much by giving you tiny samples of antibody. Additionally, though, we can go back to this page for the acetylcholinesterase antibody. And again, there we see that pretty hefty price tag for 150 microliters at like a 1 to 8 concentration. However, you'll notice there's a drop-down menu for size concentration. We drop it down and we click 20 microliters. This decreases the cost to $129. Now, for the cost per volume ratio, this is a lot more expensive. But if you're trying to spend under a certain amount and you don't need a lot of antibody, you just need enough just to get this project done, this is one of those things that you can do. If a vendor basically sells a smaller size, you can opt for that and spend a lot less. This may be particularly relevant for like those small grants that student researchers might have, for instance, as I have in the past. So do keep this option in mind. And that's it. So just to wrap this up, I hope that these ideas have helped you decide how to obtain the right antibodies for your research projects. Keep in mind that although following my tips will increase your chances of your IHC stains working out, you shouldn't expect a 100% success rate, especially if you do a lot of IHC staining. That said, the success rate maybe will be like 80% or better if you kind of keep each of these tips in mind. You should also keep in mind that there are other factors that can affect antibody staining, uh, such as things that would cause it to not work. A prominent factor for a lot of researchers is if the tissue is overfixed, meaning that it was kept in formaldehyde for far too long. This will mask targets of most antibodies and give a false negative for the staining. This is part of the reason why some vendors may suggest antigen retrieval, for instance. There are also other reasons why an antibody product may not work in your hands, but I won't really get into that here because that's a little bit out of the focus of this video. Regardless, I still believe that your success will be greater if you are careful in how you choose your antibodies from these vendors that you browse on the internet. See you in the next video.